Okay, hi everyone. This is our recording, or lecture recording, right? Oh, sorry, that's not what I meant to do. For chapter 12, which is the molecular biology of the gene. So basically, this is our chapter where we're talking about DNA replication, transcription, and translation. Okay, and there's a little bit more to the chapter as well, and I'll show you in the outline. All right, so let's first, this kind of, this is our last chapter of this molecular biology type of information. The next two chapters sort of change gears, right? In fact, they're in a separate unit. So it's chapter 12, and then the next unit we'll switch and we'll start talking about Darwin and evolution. Okay, so like I said, this is the last chapter sort of within this unit. Okay, and this is the first chapter that will be covered on the final exam. All right, so just a little bit of outline first, then a little introduction. Remember how the book always has that introductory sort of paragraph to put things into perspective for you. So we'll just do a little bit of introduction there. So the first part of the chapter talks about DNA as the genetic material and sort of how DNA was discovered as being the genetic material. Then, as I said, we'll talk about replication, transcription, and translation, and we'll talk about the genetic code. Okay, so in terms of a little introduction here, so it's titled The Genetic Basis of Eye, Hair, and Skin Color. I'm not sure it covers all of that, but it's just a really basic sort of introduction here. So it says all life on Earth contains 4 bases, right? So if we talk about the 4 bases that make up DNA, there's just 4, right? A, G, C, and T, right? Adenine, guanine, cytosine, and thymine. So these bases, right, it says they're molecules and they code for biological parts such as the proteins that make skin, bones, and eyes. Bottom line is the basis, the sequence in the DNA is what's going to code for proteins, right? And then depending on what types of proteins are produced, that will then determine what your hair color, what your skin color is, okay? It says different combinations of these bases make up genes. That's what I just said, right? So if there's a change in that sequence, it will then change that gene and then therefore maybe change the protein that's produced. So it gives an example here of one particular type of gene called MC1R. And this gene has been shown to contribute to skin, hair, and eye color, okay? And it's found in the nucleus of every cell, just like every gene, right? So when we think about DNA and we think about genes, every single gene is present in every single cell, okay? And where this gene is mostly expressed are in cells called melanocytes, okay? So there's some of the cells in skin that will become specialized, or they will differentiate and become pigment producing cells called melanocytes, and they're going to make melanin, right? And so depending on what variation you have in this MC1R gene, that will determine how much melanin is produced by these melanocytes. And depending on how much melanin is produced, that's going to affect the color of your skin, of your hair, and of your eye, okay? So there's other genes involved as well, but this is one of a gene that contributes to skin, hair, and eye color. So if you just think about skin color, right, again, depending on how much melanin is produced, that's going to cause a variation in skin color, okay? So it says here, differences in the DNA base sequence can alter gene expression, which affects how much melanin is produced. That's what I just said. So term here, gene expression, and I think I said it in the beginning as well, when we talk about gene expression, right, again, the sequence in the DNA, right, is going to ultimately code for proteins. And that's what we're going to talk about in this chapter. And essentially the gene expression part of it is what protein is made, right? Or how much of it. And so that's then going to change function, right? So if we're talking about skin color, again, based on whatever DNA sequence you have for this one particular gene, say, that's going to then cause your cells to produce a certain amount of that melanin, right? And again, depending on what amount is produced, that's going to then dictate what your skin color is, right? Or be part of that, okay? And so when we talk about gene expression, we're talking about that melanin being produced, right? That protein that's produced and how much of it then, okay? So you can have different levels. So it's not only if it's produced or not, right? It's also to what degree it's produced or how much of it, okay? So when we talk about gene expression, that's what we're talking about. Okay, so again, everyone has melanin genes, right? Everyone has melanin genes, but it depends on what that gene expression is, okay, in terms of what skin color you will have, right? Or what eye color you will have. And again, remember, there's other genes involved as well, but this is one example. Okay, so I hope that sort of kind of helps put things in perspective a little bit. So the first part of the chapter, we're going to talk about DNA, right? And the discovery of DNA. Also, remember, you have that review sheet for the final exam, and you should have that out, and you should be taking notes on that final exam review sheet and referring to it as we go through the lecture. Okay, so in terms of the first part, in terms of discovering DNA as the genetic material, now you got to bear with me a little bit in this first part. And it says this on the review sheet. I don't expect you to memorize any of these experiments. However, if I gave you a little bit of introduction about the experiment or showed you a figure, we should be able to interpret and understand, okay? All right, so basically, we're talking about in late or early 1900s, I guess. Let me just get a correct time frame because I don't know if I have it listed on the slides. So we're talking about early 1900s, okay, in terms of this is when there were a lot of scientists working on understanding what the genetic material was, right, to figure out what it was. And so it says DNA proteins were the candidates of genetic material. So remember, we think about chromosomes, right, or chromatin, we know they're composed of DNA and proteins. And so the question here is, well, which one is the genetic material, okay? And so we're just going to highlight a few sort of historic experiments. There are many, many other experiments and many other researchers that contributed to this, right? But these are just some of the sort of main ones. And also just to give you an idea of what types of experiments or research was going on at the time, okay? So we're talking about, again, deciding or trying to figure out whether it was DNA or protein as being the genetic material, right? So proteins contain 20 amino acids, and we'll talk about them, a little bit more as we move through the chapter. And these 20 amino acids can be sequenced in different ways, right? DNA and RNA, they only have 4 types of nucleotides. So based on those numbers, you may be more apt to think that proteins might be the genetic material because it seems like maybe there's a little more complexity there, okay? All right. In terms of what are the requirements for genetic material, right? They must be able to store genetic information, obviously. That molecule must be stable, right, and able to be replicated accurately during cell division and transmitted from generation to generation. Also must be able to undergo mutations, right, that provide genetic variability, allow for natural selection and adaptations, things that we've talked about sort of throughout the semester. And so researchers really showed that DNA can fulfill all of these functions. So we're going to highlight, like I said, some work that was happening, again, in sort of the early 1900s or so in terms of discovering DNA as the genetic material. And again, remember, there are many other researchers that contributed to this as well. All right, so first one we're going to mention, and I'm going to get in a little more detail in the next couple slides, but not that much. And again, we're not memorizing these things, okay? I would say, all right, here from Griffith's experiment, and show you a picture, I would then ask you questions about it, okay? So Frederick Griffith will be the first one we talked about. He investigated the virulence of a bacteria, right? Streptococcus pneumonia, okay? And basically, I feel like there could be a typo. Okay, we're good. Okay, which is pneumococcus, all right? So I just want to make sure this was spelled right. As I'm doing these, I actually find a lot of typos, more than I would if I was talking through it in class, actually, with you guys there. Okay, so he concluded from his experiments that the virulence, meaning the virulence is what effect it's going to have on, say, he did his work in mice, okay? What effect that's going to have? Is it going to kill them or not, right? How virulent is that? So So he had two different strains, one that was virulent and one that wasn't, okay? So he concluded that the virulence could be passed from a dead strain to a non-virulent living strain. So that probably doesn't mean anything right now, but once I show you the experiment and talk about it a little bit more, it might make a little bit more sense. At least I hope so. And so anyway, he called this transformation. So it was the idea that a strain, a bacterial strain that was virulent, meaning it's disease causing, it's going to cause the mice to die in this case, could basically transform another type of bacteria, another strain of bacteria that wasn't disease causing. Okay? All right. And so we'll talk about that a little bit more. So that's what that is about. And then there was further researcher by Avery, and basically a group led by Avery that discovered that DNA is actually this transforming substance. So you'll see in Frederick Griffith's experiment, he didn't know what was the transforming substance. He just knew that something did that. But the question was, is it DNA or protein, right? As I said in the beginning. And so work by Avery and colleagues did discover that DNA is the transforming substance. And like I said, there's many other researchers that contributed to this, okay? It's not only these scientists that I'm mentioning. All right, so it says DNA from dead cells can be incorporated into the genome of living cells. Okay, so in these bacterial cells, we call that transformation because, again, the non-virulent strain can actually pick up the DNA of that virulent strain, even if those cells were dead. All right, so let's talk about his experiment a little bit more. All right, so what he did was he injected mice with these two different strains that I talked about, right? These two different strains of bacteria, these pneumococcus bacteria. And one was encapsulated. So he noticed that this S strain actually when he looked at these strains on a plate, that they form these shiny colonies, okay? So that's why he called them this encapsulated S strain. And then he also had this other strain that when he looked at those strains on a plate, on a bacterial plate, they looked dull. They weren't as shiny. They didn't have this capsule, okay? So that's where those names come from. So, but what he did was he took the bacteria and he actually injected them into mice, okay? So the S strain, like I said, is virulent, meaning it causes the mice to die, okay? It has a mucus capsule, like I said, and forms shiny. That's why it's called S. The R strain, not virulent. When he injected this strain into the mice, they lived, okay? Okay. So if we look at this picture here, so like I said, I might show you this picture on the exam and ask you questions about it. So it's really about understanding what's going on here, not memorizing anything, because that doesn't help anyone, okay? So if we look here in panel A, right, it says that he injected this live S strain, right, which has the capsule, and it causes the mice to die, okay? Poor little mouse here, okay? So here's the capsulated S strain in blue. All right, now if he injects this R strain, right, that doesn't have this capsule, where you can see that no capsule here, and it's denoted in red, mice don't die, okay? This is the non-virulent strain, this is the virulent strain. If he now, so here, the virulent, right, S strain is actually killed by heat, right? So he kills the cells, the bacteria are essentially dead, right? And so when he just injects that heat-killed S strain into the mice, they don't die, right? They're fine because those bacterial cells are dead. However, in this last panel here, D, which is kind of his, I guess this sort of main point of his experiment, right? Or the picture that really depicts his experiment and what he found, right? You see here, he injected heat-killed S strain, right? In blue, you can see it in the syringe here. And then also live, oh, okay, sorry, he withdrew. Okay, sorry. All right, I just want to make sure it wasn't a typo. He injected heat-killed S strain, right, plus live R strain. Remember, the R strain is really not virulent on its own, okay? And when he did this, the mice died, right? So it suggested that somehow this genetic material from this S strain, they were dead, the cells were dead, but the genetic material was still there, right? Basically was picked up by this R strain, and then it caused these non-virulent bacterial cells to actually become virulent. It transformed them into a virulent strain. That's why we call it transformation, okay? So there was some genetic material that was transferred, to the non-virulent strain that transformed that non-virulent strain into a virulent strain. So it says here now, when he withdrew or took a sample from those dead mice, he actually saw live S strain. Why? Because again, these live cells became transformed and now they behave like that S strain. Okay? So again, I mean, kind of complicated, but hopefully we can follow that and understand what's going on there. All right, so like I said a couple slides ago, there was work by Avery and colleagues, so Avery, McLeod, and McCarty, and their experiments, I'm not going through the details of their experiments, but their experiments showed that that genetic material that was able to transform, right, that non-virulent R strain and make it virulent was DNA. Okay, so they use enzymes that break down DNA or break down RNA or break down protein, right? And basically sort of combine this with doing similar experiments like he did here. And what they showed was that when they, if they use enzymes to break down DNA, right, to basically destroy the DNA, you wouldn't be able to see that transformation occur. If they use enzymes to break down the RNA, transformations still happen. Okay, if they used enzymes to break down protein and destroy the protein, the transformation still occurred. Okay, so that's what this is saying here. It says in separate experiments to digest the substance which allowed that Streptococcus to produce a capsule and become virulent. So again, the only enzyme, like I said, that had an effect was the DNase. Okay, so that prevented that transformation because, again, if the DNA wasn't there, transformation couldn't occur because that DNA is the genetic material and that was what was responsible for causing that transformation. Okay? All right, another experiment to mention is Hershey and Chase's experiment. Okay? Again, this is all kind of still going along with some of the experiments that were important in discovering DNA as the genetic material. So here they used a virus, which is called a T-phage or T-phage. Some people say phage, some people say phage. And so a bacteriophage or a bacteriophage is a virus that infects bacteria, okay? So they used this T-phage that was composed of radioactively labeled DNA and also coat proteins, okay? And they did this in 2 separate experiments, and they infected E. coli bacteria with it. So they basically radio-labeled the DNA and the protein coat so that they then could see it, okay? So they radioactively labeled it, and then they could later on basically expose things to a film, and they could actually see where the DNA went, okay? So they discovered that the radioactively labeled DNA, right, but not the protein, actually ended up inside the bacterial cells. And then again, if these bacteriophages infect bacterial cells, what happens is that genetic material, right, can get, one second. That genetic material, right, enters into that bacterial cell. So again, their question was, well, what is the genetic material, right? Is it the protein part of the virus, or is it the DNA part? Okay, so that's what they did. They labeled it so they could tell what went inside. So it says again, the DNA, but not the protein, ended up inside the bacterial cells, causing them to become transformed. So only whatever molecule got inside the cells was responsible for this transformation. Okay, so again, it showed that only DNA and not the protein was the genetic material. So this is just another type of experiment that basically showed that DNA was the genetic material. So like I said, right, so they basically, have E. coli cultured, they add virus to it, and then they visualize by radio labeling the DNA and the protein, they visualize, right, they give it some time, right? And they basically can visualize what's happening in terms of where that DNA is going, okay? So they can see the protein's just sitting outside, but the DNA went inside the cell, okay? So it's that DNA that's the genetic material. And again, there's a lot more to say about these experiments. I mean, just like most things in this course, we can get into a lot more detail, but we're trying to just get an overview, right? And we're sort of scratching the surface of biology here. All right, so that's the last set of experiments we will talk about. Okay, again, everything that I just talked about in that last, or this first section of the lecture, were experiments or research that was important in discovering that DNA is the genetic material. Okay, and then obviously, subsequently, once we had more technology, we confirmed this over and over again. Okay, but these were some of the earlier work that really, these were really important in initially making this discovery. Okay, so now we got to talk a little bit more about DNA, right, and its structure. So, and we probably know this already. We've probably went over this before. So it contains, again, these 4 bases, right? Adenine, guanine, thymine, and cytosine. A, T, G, and C. Okay, so remember we have purines. Adenine and guanine are purines, and thymine and cytosine are pyridamines. Okay, now just a little bit to mention here something called, so another, I guess, researcher, right? Chargaff, and something called Chargaff's rules. All right. So he did work on DNA as well. And so now so again, once we knew DNA was the genetic material, then there's going to be lots and lots of people studying DNA and its structure and what it's made up of, right? Chargaff being one of them. And then we'll talk about Watson and Crick and Rosalind Franklin as well. Okay. So we know DNA is the genetic material. Now we need to know a little bit more about DNA. All right, so Chargaff's rules talk about the amounts of A, T, G, and C in DNA, or the amounts of these bases. Couple things. So the amounts of A, T, G, and C in DNA are constant among members of the same species. Okay, so same species, you have similar amounts of these bases. But the amounts of these bases will vary from species to species. So if you look at different species, you're going to see differences in the numbers or the percentage. In each species, there are equal amounts of A and T and G and C, right? And why? Because A always pairs with T and G always pairs with C. So that makes sense, right? Whatever amount of A you're going to have the same amount of T because they're always going to pair together. Okay. So this sort of initial work suggested that DNA uses complementary base pairing. So he didn't really know that at the time, okay? So it says here, each human chromosome contains on average about 140 million base pairs, okay? And so the number of possible nucleotide sequences is 4 to the power of 140 million, okay? So even though we only have 4 bases that make up DNA, again, there's a huge amount of variability or possible variability and possible nucleotide sequences because there's so many base pairs in each chromosome, okay? All right, so if you look down here at this sort of DNA composition here, right, that's what I was just mentioning. So if you look within species, right, you'll see that, again, all the members of the same species will have similar percentages here, okay? Again, one thing to notice that the amounts of A and T are very similar. The amounts of G and C are very similar. If you look at the different species here, you're going to see that those percentages of the bases will vary, okay, depending on species, right? But again, A and T, same percentage. G and C, same percentage, okay? So again, what we know now is that that is because of complementary base pairing. So we've seen a picture like this before. So just a reminder, again, these are the nucleotides, right, that make up DNA. And so if we, well, so then, and then this is actually showing you, right, the base here. So guanine, so again, adenine, guanine, thymine, and cytosine, okay? Thymine and cytosine are pyridamines, and remember those pyridamines only have 1 ring, right? And the purines, adenine and guanine, right, have 1 ring. So shorter name, what the heck am I talking about? Duh. I was going to say that doesn't make any sense. Purine, right, longer, 2 rings, pyridamine, 1 ring here, okay? So these are this is the base here. Okay, then this is showing it linked, right, to the sugar phosphate backbone of the DNA, right? So we talk about nucleotide, It's because the base right here is now linked to that sugar phosphate backbone. Okay, so don't be confused by the terminology there. Again, so in case I did confuse you, right? Pyridamines, one ring. If you look at those nitrogenous bases, right, those bases, thymine, cytosine, if you look at adenine and guanine, they contain 2 rings, okay? And so we're not just looking at these separately, we're now looking at these linked to the sugar phosphate backbone, okay? So that's why we're calling them nucleotides there. All right. Okay, so anyway, right? So it says here, the 2 nucleotides with the purine bases are adenine and guanine. So again, nucleotide refers to the base linked to the sugar phosphate backbone, okay? Just so you guys know. So they're always going to obviously be linked to the sugar phosphate backbone, as, you know, when we think about them as being part of DNA, okay? Or RNA as well, right? Okay, so now we got to talk a little bit more about some work, okay, in terms of the structure of the DNA. So I guess you can kind of think of char graph, what I just mentioned in terms of the composition of the bases, right? Now this is really talking about the structure and how that DNA is put together, okay? So first, I guess scientists to talk about is Rosalind Franklin, though most people probably don't know that much about her. But she was really the first one that really was studying the structure of DNA, and she was using X-rays, right? So basically, remember how we extracted DNA and you had that goopy kind of, we precipitated it out, right? When I did the DNA extraction lab, what she basically did was then shine an X-ray through that and then basically get the pattern of the sort of the diffraction of those rays on a film, right? And so that's how she sort of was looking at the structure. And so, so this is what this is saying here, right? These fibers, once you separate them, like, again, similarly to the way we did the DNA extraction, you basically can get this X-ray diffraction pattern. I'll show you a picture in a second. And when you look at those photographs that she produced from this X-ray diffraction, it looked like, right, DNA is a helix. And then it also looked like there was some portion of it that was repeated, okay? So supposedly a colleague of Rosalind Franklin actually showed her these micrographs, or they're not micrographs, these photographs, right? These X-ray diffraction photographs to James Watson, and he apparently was the one who really grasped the significance of that pattern and what that meant for the DNA structure. Okay, so it's almost like she did the legwork, did the initial work, but they really, Watson and then his colleague Crick, really interpreted that information and came up with the model, right? That DNA double helix model that we think of today, okay? And so usually here, Watson and Crick, they're the ones who got the Nobel Prize, and you sometimes don't hear too much about Rosalind Franklin. Anyway, that's what this is just showing you or illustrating it, right? Here's your, basically you have your X-ray beam, here's your DNA, right? So again, you precipitate it out, you can crystallize it, and then send X-rays through. Look at that diffraction pattern, and this is an example of what she saw, okay? All right, so like I said, then Watson and Crick really took those photographs and really interpreted them and came up with this model, okay? So this Watson and Crick model was developed in 1953, all right? And it still holds true today, okay? So like I said, they did receive the Nobel Prize in 1962 for this model. Okay, so that double helix structure is similar to like a twisted ladder, okay? The sugar phosphate backbones make up the sides of the ladder, right? And the bases make up the rungs, right? And those bases are actually hydrogen bonded to each other. The 2 strands are also anti-parallel, okay? You always have to, and I'll show you what that means in a second. You always have to read DNA information in a 5' to 3' direction. So just like when you're reading a sentence, you read it from left to right. If you read it from right to left, it will not make any sense, right? So same thing with DNA. It has to be read in that 5' to 3' direction. Because remember, the information in the DNA is stored in its sequence, right? If you went the other way, you're totally changing the sequence. Just like, again, if you're reading a sentence from right to left, you're changing the sequence of those letters, and therefore they don't make up any words, okay? So it says here, so DNA is always replicated in the 5' to 3' direction, and we'll talk about that once we talk about DNA replication. Complementary base pairing ensures that a purine is always bonded to a pyridamine, okay? A with T, G with C. And like I said, they received that Nobel Prize in 1962. So this is just showing you a picture of their early version of this DNA double helix model. If we look at this cartoon version here, a couple things to point out, right? This sort of twisted ladder structure. Again, this is the sugar phosphate backbone, right? You can see this here, sugar molecule, phosphates, right? That link together to form that backbone. And the rungs are formed by the bases. The bases are joined together by hydrogen bonds. Again, we talk about complementary base pairing, right? G always pairs with C, A always pairs with T. Okay, a purine and a pyridamine. The other thing I mentioned was this anti-parallel structure of DNA, in that you have, and the fact that it has to always be read in the 5' to 3' direction. So, you know, going this way here, okay, right, or this way. So anyway, 5' to 3'. So if you again look here, it's anti-parallel. What we mean by anti-parallel is this strand is 5' to 3' going this way. This one, 5' to 3' going the other way. Okay? All right. So the next part of this, we're going to talk about DNA replication. So remember, we've mentioned DNA replication, right? In our chapter when we were talking about the cell cycle and mitosis. Right? We said that DNA has to be replicated before the cell can divide. Okay? So what's DNA replication? It's just the cell making an exact copy of the DNA. Okay? So another term here, semi-conservative replication, it means that each strand of that double helix, right, serves as a template. So bottom line is, when you're replicating the DNA, you're going to have one old strand and then a new strand that's going to be made, because it's made via complementary base pairing, okay? So that's what we mean by semi-conservative. So it says here, the parental molecule, one strand will serve as the template for the new strand in the daughter molecule, okay? So I'm going to show you some figures to show you what we mean by that. So again, remember, the DNA is in a double helix, right? If we talk about the structure of the DNA, in order for that DNA to be replicated, what has to happen is those two strands have to separate, right? And so we call that sort of point where they separate a replication fork, okay? And again, if you look at this, the 5' to 3' direction, right? Something to pay attention to here. So 3', right? The blue's the parental strand. This is what's going to be used as a template to make the new green strand, right? So here's 3' on this end, 5' up here. If we're talking about this being made here, right? This is being synthesized in the 5' to 3' direction, okay? So again, and you can see the green, again, these new incoming nucleotides coming in that are going to pair via complementary base pairing. So what this is showing you, again, here's your DNA that you're starting with that needs to be replicated, okay? Each one of these strands will serve as a template for the new daughter strands that are being produced, okay? So the green is the new. So that's what we mean by semi-conservative. If you look at the newly replicated DNA, it's going to have one parental strand from here and one newly synthesized strand. Same thing, right? One parental strand, one newly synthesized strand. So now you're ending up with two copies of that DNA. Okay, now we have to talk about the details of this, right? And again, you may have been introduced to this at some point, but the one thing to mention is there are many, many proteins and enzymes that are involved in DNA replication, okay? So it's not just like there's one single protein that replicates the DNA and that's it. There are many proteins involved, all of which have a specific job, okay? So I'm going to start to list some of these here. So it says here, unwinding or separation of the two strands of the parental DNA is done by that DNA helicase. So remember I mentioned that replication fork, right? And that unwinding and that separation of those strands. Well, there's a particular enzyme called DNA helicase that's responsible for that, okay? Like I said, there are many other enzymes and proteins that are going to be involved in DNA replication as well. So single-stranded binding proteins, we can abbreviate them as SSB, they will attach to the newly separated DNA and prevent that helix from reforming, or prevent those strands from coming back together, because it has to stay separate so that those two strands can serve as the templates, right? Okay, it says here complementary base pairing between a new nucleotide and a nucleotide on the template strand, right? What's helping with that part of it? So DNA primase will actually place short primers on the strands to be replicated. So DNA replication has to start with a primer, okay? And all a primer is just a short piece of DNA that's going to bind to the template strand, okay? Then the main enzyme that actually is going to synthesize the new strand, okay, is polymerase, DNA polymerase. So it recognizes the RNA primer. So, okay, so DNA primase puts these RNA primers down, and then the polymerase comes along, recognizes that, and basically adds to that, and basically synthesizes that new strand, okay? Again, it has to be synthesized in the 5' to 3' direction, okay? Now, because it has to be synthesized in the 5' to 3' direction, okay, the two strands are replicated differently. You have a leading strand and you have a lagging strand, right? On the lagging strand, you actually have replication that sort of occurs in fragments, okay? And so it says you basically need DNA ligase there as well, because it has to join these fragments together. I'm going to show you a picture and explain these again with a figure. Okay, I already showed you this on the last slide, though. Each daughter DNA molecule will contain one old and one new strand, all right? Okay, so we're going to talk a little bit about, or I'm going to show you a picture, eventually, okay? I guess we got to get through this stuff first. So keep in mind these proteins and enzymes are going to come back again in the figure that I show you, and I'll point these out again. And actually, just by looking at the figure, you really can see what each one of these proteins or enzymes are doing, okay, what their job is. Now, if you talk about replication of DNA between prokaryotes, right, prokaryotic cells like bacterial cells versus eukaryotic cells, it's going to be different, okay? So for prokaryotes, one thing a major difference is that bacteria just have a single circular chromosome, right? A singular circular loop of DNA. And so replication is a lot easier. Okay, so replication just moves around the circular DNA molecule in both directions, and then it just produces these two new circles, okay? And it begins at something called an origin of replication, okay? So kind of, right, that replication fork, what I sort of pointed out, we can think of that as sort of the origin of replication. Takes only about 40 minutes. Cell divides every 20 minutes. Basically, a new round of replication can actually begin before the previous round is completed, okay? So bacterial cells replicate their DNA really quickly, divide really quickly. They don't really have much else to worry about, right? If we talk about eukaryotic replication, it's more complicated, okay? We have linear DNA, right? So it says DNA replication will begin at numerous points along the linear chromosome. So it's not just going to be at one origin of replication. There's going to be many in eukaryotic cells, okay? I showed you, or I talked to you about that on a couple slides ago, that it has to unwind and unzip, right? And then each old strand serves as the template for the new strand. We already said this too, and I already mentioned polymerase. That enzyme, DNA polymerase, is the actual enzyme that adds the new nucleotides, right? That basically synthesizes that new DNA. Okay, so like we said, in eukaryotes, you have these multiple origins of replication, and what they kind of look like is these replication bubbles, okay? And so they'll sort of spread bidirectionally, right? They'll kind of start doing their thing and replicating until they meet, okay? So it would not be possible to replicate the entire genome, say, of a human cell by just starting in one spot, okay? So it's necessary to start in many different places. All right. So I already said this, replication is semi-conservative. Sorry, some of this is redundant. Okay, we already said this, right? One original strand is conserved in each daughter molecule. Remember, one of the parent molecule strands, or each one of the parent molecule strands, serve as a template for the newly synthesized strand, okay? One other thing to mention here is that DNA polymerase is actually unable to replicate the extreme ends of chromosomes, okay? So there's something called telomeres, which are just repeat sequences of DNA that exist at the extreme ends, because these aren't going to be genes at these ends. They're just, again, repeat sequences that sort of protect the ends, okay? There's a lot more to say about telomeres, but that's for another time. All right, so if you look at prokaryotic versus eukaryotic replication just in general, right, in terms of kind of the origin and how it's starting. Remember, in bacterial cells or prokaryotes, it's just one circular DNA molecule, okay? Super easy. Just one origin of replication, and it just goes around and makes a new strand, okay? Super easy, right? It's just occurring, right? Just goes this way and this way until the entire thing is replicated. In prokaryotes, again, in eukaryotes down here, right? So this is prokaryotes up here, eukaryotes down here. It's linear. The DNA is linear, okay? Again, you don't just start in one spot. You start in many spots. And so if you want to I talked about the replication fork kind of being right here. This whole thing you can call a replication bubble. So if you would look at DNA being replicated under the microscope in a eukaryotic cell, you'll see many replication bubbles, okay? And so again, it'll be bidirectional in terms of being synthesized. You can see this here until they meet, and then that's it. That's done, okay? And then again, when you finish, you're going to end up with 2 new DNA molecules, right? And each one of them will have the parental strand and a newly synthesized daughter strand, okay? Same thing that you saw here, but again, much more simple process in prokaryotes because there is just one circular chromosome. All right. Now, as I said, I was going to bring back a picture that showed you these proteins / enzymes that I mentioned, okay? And then there's also a table that describes the function of them as well. So again, something important. Maybe I'll show you this, right, on the exam and say, hey, can you tell me what the function of DNA polymerase is? And you'll have a clue because you're looking at it here. Again, not about memorizing, about understanding. So again, this is just showing you one replication fork, right? So it's showing you DNA helicase at the replication fork. Remember, DNA helicase will unwind, right, this parental strain. So if you want to look here on the table, it'll give you a little bit of a function there, okay? Separates the strands, right, and sort of unwinds or separates the strands. So now you have each strand separated so that each strand can act as a template, okay? If you now look at this strand here, this is the leading strand, okay? Here's your DNA polymerase, and you can see that this is synthesized continuously, okay? Because it's being synthesized in that 5' to 3' direction. I mean, to be honest, this figure probably should show the 5' and the 3' direction here, but it's not. So, I mean, don't worry too much about it. But remember, the reason why this can be synthesized continuously is because, again, that DNA is synthesized in that 5' to 3' direction here. Here, it has to be sort of synthesized, it has to be synthesized this way because, again, it can only be synthesized 5' to 3', but the direction of the fork is going the other way, okay? Again, if that kind of gets you confused or whatever, I think maybe just kind of take this at face value for now. Like I said, in cell biology, we get in a little more detail about this. But again, the reason why the lagging strand has to be produced in fragments, these Okazaki fragments, is because it has to still be synthesized in this 5' to 3' direction, okay? So again, this synthesis is going in the same direction, sort of as that replication fork here. Here, you can see it's going against because, again, it has to be synthesized in that 5' to 3' direction. So it's synthesized in these fragments, right? These Okazaki fragments. And then you need another enzyme called DNA ligase that comes in and joins the fragments together. In the end, it's going to look the same, right? You have a parental strand with a newly synthesized daughter strand. If you notice these single-stranded binding proteins, right? These are going to keep the strands separate. Okay, here again, DNA polymerase. DNA polymerase is the main enzyme that is synthesizing that new strand. That's what's actually adding the nucleotides in based on complementary base pairing. Another thing here, you notice this RNA primer, right? In purple here, DNA primase. The DNA polymerase DNA replication has to start with this primer here. So the DNA polymerase would not be able to add the nucleotides and would not be able to synthesize the new DNA if the primer was not here, okay? So DNA primase adds this RNA primer, which is just a short piece of RNA, okay? That binds here, again, via complementary base pairing. And then now the DNA polymerase can come along and add those nucleotides and sort of extend it, okay? So I think I pointed out all of the proteins here. Again, leading strand, we should be able to pick out leading strand versus lagging strand. Lagging strand has to be synthesized in these fragments because, again, synthesis has to go in that 5' to 3' direction. Those fragments are called Okazaki fragments, named after the Japanese scientist who discovered them. DNA ligase on the lagging strand has to come in and join these fragments together, okay? So hopefully that makes sense. Again, remember the purpose of DNA replication is to replicate the entire genome, right? The entire DNA before the cell is going to divide. So if we're talking about in our bodies, we're talking about cells that are undergoing mitosis, right? During times of growth and repair, the DNA has to be replicated. Remember, it's replicated in S phase of the cell cycle. Well, now here are the details in terms of how the DNA is replicated, all right? In bacterial cells, remember that replication is a lot more simple, right? Because it's just one circular chromosome. And I'm not even going into where those proteins fit into play. But again, in terms of what's actually happening at that replication fork in the bacterial cell, it's going to be similar to here, okay? So what I want you to know is, again, not memorize these, but you should know the function of these proteins / enzymes, right? Whatever, I'll just say proteins in general. A lot of them are enzymes. So, but not all of them. So I want you to know the function of these. But again, if I'm going to ask you the function, I mean, sometimes I'll ask you about function in a multiple choice question, but in an essay question, I'm not going to just ask you to list the functions of these proteins. I would probably show you this and maybe ask about the functions or ask questions that pertain to this figure here. Okay, so a lot of times on the final exam, I do use this figure in one of the essay questions. All right, so I'll leave it at that. Again, purpose of DNA replication replicates the entire genome, replicates all the DNA. And when we talk about actually how that happens, right, when you get the newly replicated DNA, so you start with one copy of DNA, you end with 2 copies. Within those 2 copies of the DNA, you have one parental strand and one newly synthesized strand, okay? All right, and again, this table gives you the definitions of those proteins that I just went through on this figure. All right, in terms of accuracy of replication, it's pretty accurate, actually. So, I mean, you can imagine it's probably pretty important that the DNA polymerase is accurate, right? But it still does make some mistakes. So it makes a mistake about once every 100,000 base pairs, which is an issue. You don't really want it to make any mistakes. However, the DNA polymerase is actually also capable of correcting its errors, okay? So because it has this ability of identifying an error, meaning it puts, so there's an A in the template strand, and instead of putting a T, it puts a C, right? That would change the sequence, right? And basically induce a mutation. However, it has the ability to recognize that mistake and fix it and replace it, okay? So with that ability to proofread or to fix its mistakes, it actually, if you talk about bacterial DNA polymerase, its error rate is only about 1 in a million instead of 1 in 100,000, okay? So the ability of proofreading or correcting its mistakes greatly increases the accuracy of the DNA polymerase. Okay, so if I asked you what the main enzyme is in DNA replication, you should right away say DNA polymerase. Okay, that's the enzyme that's going to be adding the new nucleotides, right? It's actually looking at the template saying, okay, there's an A there, I put a T there. Okay, so that's what's actually synthesizing that new DNA. Of course, it needs all those other proteins and enzymes that I talked about as well, because otherwise the replication wouldn't happen. But DNA polymerase is really the main enzyme. Okay, I'm going to stop there for this part of the lecture, and then I will pick up part 2 of chapter 12 with the genetic code. Okay, so we talked about the discovery of DNA as the genetic material. I gave you some examples of some important experiments that happened in the 1900s, early 1900s. And then we talked about the process of DNA replication, okay? Again, think about the function of replication first. It is that you are replicating the entire genome. When are cells doing this? They are replicating the entire genome before they divide, okay? So bacterial cells do it all the time, right? That's what they're gonna do in terms of reproduction, right? They're gonna replicate their DNA and divide. For our cells, we think of mitosis, right? Growth and repair. Cell needs to replicate its DNA before it divides and goes through mitosis. All right, so the next or the last part or the second part of the chapter, we'll talk about now going from DNA, right, to RNA to proteins. I talked a little bit about gene expression in the beginning of the chapter in that introduction. Now we're going to talk about gene expression. Okay, so I'll leave it there and I'll pick up with gene expression in the second part.